Hello from the International Workshop on Non-Hodgkin Lymphoma in Italy in 2015. We just had a very interesting session on the biology of diffuse large B-cell lymphoma and how it impacts on future concepts in the treatment of diffuse large B-cell lymphoma. And Randy Gascoyne gave us the lead, how we should do this, and I think which was quite interesting. You set an order, what we have to follow. First order decision, second order decision, and maybe a third order decision. Perhaps you explain this a little bit to
Sure. So I think what everybody in the world has now come to grips with is that large cell lymphoma is very heterogeneous at the molecular level. And as far back as 2000, we recognized using gene expression profiling that there were two dominant subtypes of disease, one known as GCB, or germinal-center B, and the other one activated B cell type, or ABC. And now it's taken many years of a lot of frustration with immunohistochemistry and a whole lot of different algorithms that never produced very robust data and have issues about reproducibility and accuracy as it relates to determining those molecular subtypes. And now our group in Vancouver, but other groups, have developed different strategies, different sort of technological platforms that are of value in helping to make those distinctions between GCB and ABC. And what is really great about them is they appear very accurate, they're extremely robust, they're easily done, they're done at a very, very small expense, and they can all be done using the material that is common to every patient with cancer, including DLBCL, and that is a formalin-fixed piece of tissue that can then have RNA extracted and the analysis run. And so the point that I was really trying to make is that to a large extent it's an initial first step that takes us down a road of trying to achieve precision medicine. So if we can accurately distinguish the two dominant molecular subtypes, we can then think about the kinds of tests that then could be appropriately applied at that point to further refine the diagnosis and to help inform on therapeutics. And the example that I used for Dr. Yunus in particular was if in fact we could tell him a case was absolutely GCB, we could then decide that that's a case that needs MYC and BCL2 FISH. And if both of those are done and they're negative, the 5-year survival for that group is probably 95% by simply doing a cell of origin and 2 simple FISH tests.
So the material is available, the method is available, but how difficult and how reproducible is the nanostring technology in the hands of an unexperienced, let's say, pathologist?
Sure. So I think what nanostring would like to see in the world is they're interested in what is called a distributed model. They would like to place their instruments all over the world in predominantly major labs. I think that if the instructions are followed and SOPs are created and followed well, the technology is extremely robust. You might remember that in that first paper in Blood last year that we published, we extracted RNA in 2 centers, one in Frederick, Maryland, and one in Vancouver, British Columbia. And in both places, the nanostring assay was done, and the reproducibility was 99.9%. So that exercise wasn't actually done with the same pool of RNA. It was simply the same case extracted in 2 different centers and run on the platform. So I think the platform is extremely robust, and in part the technology benefits, in contrast to what's called RT-PCR, is that nanostring is a direct digital readout of message that involves no enzymes, no amplification steps at all. And so I think its linear range, its dynamic range is unbelievably good, and the accuracy is unmatched almost.
What struck me a little bit, you said that the nanostring or Lymph2X assigns the cell of origin maybe even better than the good old gene expression profiling and fresh tissue did.
Sure.
How do you come to this conclusion?
So basically looking at the data over the last several years that we've been doing this technology. And I'll see if I can explain this. It's a little bit complicated. So both assays are essentially a nucleic acid hybridization. And there are multiple steps in doing a genome-wide, so-called unbiased gene expression using API. But at the end of the day, you're hybridizing 2 nucleic acids. And there are vagaries with that technology. And I think that they are reduced somewhat by a more simple assay with a lower content of genes that are valuable than assessing 54,000 probes on an Affymetrix U133 array. And when we compared the data, remember the only way we could do that original study was to assume that the Affymetrix was the gold standard. But in fact, when you look at the Lymph2CX data, or you look at replicate analysis using either of those 2 platforms, the far more reproducible, accurate platform is the nanostring assay, not the Affymetrix U133.
Okay, very interesting.
Yeah.
And then the second part, Kieron Dunleavy showed us examples how this is already translated into ongoing clinical trials. Kieron.
Yeah, so the question is, we now can identify if people have the ABC or GCB subtype, and over several studies, the ABC subtype is associated with a much worse outcome. And the big question is, how can we augment cure in that subtype? We now understand a lot of the critical pathways in NF-kappa B activation and have identified a number of novel mutations over the last few years. And there are lots of promising agents in development for ABC large cell. I think 2 of them that are particularly promising are ibrutinib, which is a BTK inhibitor, and lenalidomide, which inhibits IRF4 and interferon signaling. And both of those have a response rate of between 30 and 40% in patients who have relapsed and refractory ABC large cell lymphoma. And they're actually both being studied in upfront studies in newly diagnosed patients, obviously, with ABC lymphoma. So I think the results of those studies, which will be available in hopefully 2 or 3 years' time, will be very interesting, and hopefully they will augment the cure rate in ABC lymphoma, which does not do as well as the GCB subtype.
And then you reported the design of a trial of another type of mostly ABC or cell of origin lymphoma, that's the primary CNS lymphoma. And I think that's a very interesting approach, and perhaps you describe how you selected the drugs included in this combination for primary CNS lymphoma.
Yeah, so most or almost all primary CNS lymphomas are of the activated B cell subtype, and when you actually look at the proportion of these cases that have got, for example, MYD88 mutations and CD79B mutations, they're very, very high, much higher than systemic ABC lymphomas. So targeting B cell receptor signaling might be particularly beneficial for this group of patients. So we designed a study where we incorporated ibrutinib, and we used drugs that are effective in systemic large cell, and where we felt they crossed the blood-brain barrier, we included them, and then otherwise we modified them or used drugs in the same class to design a regimen.
And the so-called TEDdy regimen, I would guess that it's much less toxic than the high-dose methotrexate, high-dose RC, plus autologous stem cell transplantation. Is this right?
Yeah,
you probably already have information on the toxicity.
It's not been compared, but I think overall it is less toxic. One side effect of the regimen, we use Doxil, and we use pretty high doses because I think of all the drugs in the regimen, that's probably the one that is most important for curing large cell lymphoma, and that causes hand-foot syndrome. So we have modified our protocol to deal with that, but that's one of the big side effects that we've seen.
What's the dose of the
So we started with 50 milligrams.
And
we have a dose reduction scheme according to toxicity. So if we see hand-foot syndrome, we have a very low threshold for reducing the dose.
Okay, I think we covered the most important points that have been discussed in this session, and I thank you for coming to this interview.
Great, thank you.
Thank you.